Life seems to be ruled by chance. Where we are born, what we become, who we meet, how we die. Novozymes Prize winner Gunnar Von Heijne has devoted his life to bringing order into the chaos that seems to characterize life. He has become an expert in predicting how life unfolds and how it folds. Proteins are the building blocks of all life. Most proteins fold correctly, but when they misfold, diseases can develop. Particularly important is how the proteins are embedded in the cell membranes, where they serve as ion channels, pumps and receptors to transport ions, molecules and cellular messages.
So it turns out that about half of all the drugs that we're using, that you can buy in the pharmacy, actually bind to, are directed against membrane proteins. But out of the total population of proteins, maybe 30% are membrane proteins. So they are, unproportionally important.
Gunnar Von Heijne receives the Novozymes Prize because he has made so many significant contributions to our understanding about membrane proteins, and which is so important for so many areas, both biotechnology and basic science.
Gunnar Von Heine's very theoretical approach in often highly experimentally oriented biology has created great innovations. But why this career path? Chance, he claims. He grew up in the 1960s, the heyday of science and technology and the space race.
I remember getting a couple of books for a Christmas present from my grandmother. And one, it was a popular science kind of picture book about mathematics. And I still remember a picture of a mathematician lying on his back in his office on a couch and sort of looking into the ceiling and thinking deep thoughts about mathematics. I must have thought that that's the way to spend life.
The inspiration for what would be his lifelong interest came by serendipity. As a PhD student, he took an evening class in French. And because he liked French, he subscribed to a popular French science magazine.
There was a little cartoon in this paper, and it didn't make sense to me because it had some strange interactions between a protein chain and a membrane that I thought couldn't be right. So I looked up the original paper and read it a bit more and realized that, here's another thing that maybe we can make a simple theoretical model of.
The cartoon was from a paper that later led to Gunter Blobel winning a Nobel Prize. Blobel picked up on Gunnar Von Heijne's paper and invited him to a meeting in the United States.
Everybody was there. This was kind of when the field started. And the people who then drove this field for the next 20 years were all young scientists and were there at the time. So this was really, the opening of a completely new world for me.
The hypothesis at that time was that the secreted proteins all start with a short sequence of amino acids that serves as an address label to tell the cell if a protein should be secreted from the cell. Gunnar Von Heijne constantly tried to improve his models, but he felt stuck.
But again, I was lucky, and the element of luck this time was that right about this time was when people were starting to sequence DNA. I had the idea that maybe I can just collect some of these little sequences and compare them to each other and see if there's anything in common.
What emerged was an underlying design of these little address labels, the so-called signal peptides. Today, all the big pharmaceutical companies use the findings when producing protein-based medical products. Then he got interested in membrane proteins.
In membrane proteins, you find segments of very non-polar or hydrophobic amino acids that eventually end up spanning the membrane. What's different is that positively charged amino acids are much more abundant in the loops on the inside than in the loops that face the outside. So this became known as the positive inside rule.
With this rule, it's possible to predict how the protein will end up in the membrane, and particularly also its orientation and hereby its topology. And it also gives very important information about its function. And so that is one major contribution.
Gunnar Von Heijne's career took an unexpected turn. In 1995, Von Heijne and his family spent a winter in Los Angeles visiting Bill Wickner's laboratory at UCLA.
When we came there, he said, do something different, forget about your computer for a bit and do some lab work. You can do anything you want in my lab. And I had done, because I was trained as a chemist, a lab wasn't completely alien to me. So I thought maybe, yeah, why not?
The idea was simple.
Maybe we could move some positively charged amino acids from this loop that was on the inside, take them away from there and put them on one of the termini of the protein that was normally on the outside.
If Gunnar von Hein could invert the charge balance, he could not only predict the structures, but also change them.
Probably one of my best days in science was when I could develop the first gel, seeing that indeed this engineered protein had inserted backwards into the membrane. So what used to be on the outside was now on the inside and vice versa.
This experience moved von Hein's research in a direction no one could have foreseen. He became an experimentalist.
One of the best pieces of luck I think I've had throughout my career. It's great to do theory, but to actually seeing things working in the real world and designing an experiment, and the system you're studying actually behaves as you thought it might behave, or perhaps even more interestingly, doesn't behave as you thought it would behave.
As a professor at Stockholm University, von Hein's insightful research and dedication was rewarded when he came across another very interesting aspect of membrane proteins.
Usually the part on the outside has a different function than the part on the inside, but we happened to stumble across a class of membrane proteins that actually insert both ways. How could nature design a protein of that kind?
In a series of landmark publications, Gunnar von Hein and his colleagues showed that dual typology proteins are actually very common, and especially among proteins that can transport small molecules in and out of the cell. Cells use this mechanism to neutralize the effect of toxic compounds.
I think what makes Gunnar Von Heijne a great scientist is that he is very interested in some fundamental questions, and then he has an ability to really think out of the box in terms of finding completely new ways of doing this, and then also the fact that he has mastered what relatively few people do, to combine theoretical studies with cleverly designed experimental studies.
I think in a way I was lucky, because there weren't that many people that came from that background. So it meant that at least the early work, there was very little competition. Nobody else was crazy enough to do these things.
According to von Hein, life events very often happen by chance, pushing you one way or another, or you choose to go one way over another for no particular reason.
On the other hand, science, maybe from a deep psychological point of view, doing science is kind of trying to get in control, trying to understand enough about the system that you can predict what's going to happen, that you can control what's going to happen.
Although Gunnar von Hein has spent his whole career making predictions, he had not predicted he would receive the Novozymes Prize. However, he knew from his many years as a chair and secretary of the Nobel Committee for Chemistry how to react.
You could say that there are essentially three types of reactions that I've experienced. One is people who don't believe that they've been awarded a Nobel Prize, and they think somebody's pulling their leg, making a bad practical joke. Then there is the other extreme, people who have been expecting this for so many years and they're almost a little put off that it took so long. And they just say, oh, okay, thank you. and finally, there is the majority, which are just totally taken aback and go quiet. I probably fell in the third category, I think, in my little way when I got this phone call from the Novozymes Prize.